Hello everyone, my name is Alex Surnov, and today I would like to present you a very short talk on the topic of that whole area of biomedical research that was appreciated by the Nobel Prize committee in this season, the year 2025. The reason why I decided to record this video is because the topic is very much close to my professional interest. I have been working in the same area of research starting from 2008, basically from the periods when I was an undergraduate student. And I truly appreciate the whole theory that was developed in this field, the breaking through discoveries that eventually led to this Nobel Prize, and just the whole beauty of the biological structures and phenomena that this field covers. So obviously I cannot talk about all the findings in the field with a full description of their proofs, because that will take several lectures, I don't know, 2 hours long each, when doing it from scratch. Therefore, I will restrict myself by just describing you the key views that the international scientific community has on this question, on this whole field, without going deeper in the methods, how these views were first proclaimed, then confirmed, and then solidified as scientific truth with no doubt. And obviously my today's talk will be very simplified. I will leave aside some hidden exceptions, some minor effects that don't affect the whole picture considerably, and so on. All right, so our discussion today will lie in the area of immunology. That's the branch of biology that studies the processes that go on when our body wants to fight against potentially pathogenic invaders like bacteria, viruses, so anything foreign, anything alien to the body. So which levels of defense it encounters and how the body effectively fights against it or reacts on it. Let's recall those very simple basic things. Our body is composed of cells. Each cell can be approximately thought of as a little ball with a characteristic diameter of, let's say, 1/100 of a millimeter. So it's really a tiny structure, but physiologically pretty self-sufficient. Each cell has a full set of DNA, which in principle encodes all structures in the body. But obviously different cells keep different regions of DNA silent and express only those of them that are particularly needed for the physiological function that the cell is aimed to execute. For example, the cells of our muscles express those fragments of DNA, those genes that enable them to contract and relax, and therefore allow us to move our limbs. The cells that compose our neural system express those genes that allow them to fire electric signals and communicate with each other and therefore produce somehow our cognitive activity. And in line with this, these cells that compose the immune system produce multiple proteins, express multiple genes that aim to identify, isolate, and specifically react on foreign proteins that the body encounters. One of the very initial levels of our immune defense is built of the so-called T cells, T lymphocytes. T like a letter, T, which is derived from the name of the organ where these cells develop. The organ is called the thymus. Thymus lies on top of our thorax and actually serves as a hub for producing the T cells. So each T cell carries a protein-based receptor on their surfaces, which is of random structure. So one T lymphocyte has a protein, a T cell receptor, so-called, with a given structure. Another T lymphocyte in the same body, in the same individual, has a T cell receptor with a slightly different structure. Yet another T lymphocyte has yet a different receptor, and so on. There are groups of T cells that have the same receptors. Those groups are called clones. A clone is a group of T cells that shares the same receptor. And only one receptor can be expressed by a single T cell. There are no T cells that express two, or almost no T cells that express two variants of TCR. But the whole variety of TCR variants expressed by the whole population of the T cells in the body is tremendous. It's estimated as 3 times 10 to the power 8 or even higher than that. So it's a really huge number. And the benefits of this system is that the variety of T cells is so huge that virtually any alien protein that a body encounters can find a receptor that is cognate to it and only to it, or almost only to it. Meaning that no matter which pathogenic agent, which pathogenic protein gets into our body, if it travels through the body for long enough time, the it will inevitably find a T cell whose random receptor fits to this pathogenic protein, antigenic protein, binds to it specifically, and therefore allows the T cell to initiate a specific immune reaction only against this protein. And another protein will eventually encounter another T cell with another T cell receptor that is specific to that other antigenic protein just by chance. Okay, great. Such a layout allows the body to initiate immune reactions specifically and not massively against whatever else in a targeted manner. But this layout as well has a intrinsic disadvantage, meaning that if the structures of T-cell receptors are indeed random, it is pretty easy to imagine that some of those will be reactive against the proteins that compose our own bodies. We are built of proteins, cells are full of proteins, our skin is composed of, skin proteins, our mucus and intestine is composed of their proteins, and if the variety of T-cell receptors is truly random, well, some of them will be necessarily specific against the body's own proteins if the variety is large enough. In order to avoid autoimmune reactions, the T cells undergo some sort of selection in the thymus, meaning that those clones of T cells whose TCR variants are by chance specific against at least something naturally present in our body become eliminated. Those T cell clones just get exterminated and deleted from the body, essentially killed by the cells inside the thymus. And only those T cells, in theory, in principle, leave the thymus and populate the peripheral immune system and start actioning, whose T cell receptors are specific to whatever else but the proteins of our body. However, not everything is so simple as that, because some portion of autoreactive T cells also escape the thymic selection, just because this process is not 100% efficient. And they populate the body as a minor population, as a minor percentage of all T cells, but they are there, they're present. And those T cells bear a potential threat, because it's them who, in principle, initiate autoimmune conditions like type 1 diabetes, like multiple sclerosis, rheumatoid arthritis, lupus, all those inflammations against our own tissues are one way or another initiated, enhanced, or mediated by the autoreactive T cells. Okay? In order to keep these cells under control and in order to not allow them to run their autoreactive activity, there is a subpopulation of T cells, some kind of guardian T cells, controller T cells, which are officially called regulatory T cells. The regulatory T cells do not promote the immune response in contrast with other T cells, but instead they suppress the immune response. And they can originate in the body in two different ways. One subpopulation of the regulatory T cells originates straight in the thymus. And they are characterized by an enhanced specificity to the proteins of our own body. They are specific to ourselves, but they are not harmful. They do not promote the immune response, as I just said. Instead, they tolerate our body against the self-antigens. Is exactly their specificity to self allows them to do so. Another subpopulation of regulatory T cells is called the peripherally induced regulatory T cells, and they are produced from the regular T cells, from the T cells that passed the selection in the thymus and eventually populated the body. And their repertoire of T cell receptors is not that different from the repertoire of the conventional T cells, of the T cells that exert the pro-immunogenic function. And the function of those peripherally induced regulatory T cells is different. They induce and maintain the tolerance against food antigens, against some proteins and peptides that are knowingly not harmful and that are evolutionary beneficial to ignore immunity-wise, to evolutionary beneficial to not induce immune response against. So, as I said, those T cells are converted from the regular conventional T cells, and usually it happens in such organs that contact a lot with a foreign yet non-harmful environment, like our intestine, our lungs, our mucosal surfaces in the mouth. Some of those T cells are induced during pregnancy because the fetus is a histologically foreign formation as well. And basically that's it. So there are two subpopulations of regulatory T cells, and regulatory T cells are by themselves a very small portion of all T cells, but very important ones. Without them, the T cells that are mistakenly autoreactive, that mistakenly have avoided the thymic selection, would eventually go hyperactivated by the proteins of our own body and lead to severe systemic inflammation, severe autoimmune reaction. Okay, that's the science, an overall science behind that, behind the field. That's the branch of immunology in which I have been working until recent times. And now a couple of words about the current Nobel Prize winners. So the Nobel Prize in Physiology and Medicine of 2025 was given to Shimon Sakaguchi, Fred Ramsdell, and Mary Brankov. Shimon Sakaguchi led the research in the early 90s. In 1995, there was that paper in the Journal of Immunology that described regulatory T cells first. They isolated those cells. They proved that those cells are immunosuppressive, and they initiated essentially the cell level study of the T cell peripheral tolerance. Fred Ramsdell and Mary Brankov, in turn, discovered discovered the gene and the mutation in this gene that leads to absence of the regulatory T cells. So as I said previously in the very beginning, every cell type has one or several genes or proteins, genes that are expressed specifically in this cell type and that ensure the identity of this cell type. Those genes are called master regulators. So the regulatory T cells indeed have such a master regulator gene. In mice, it is almost a unique master regulator, sufficient and necessary for a T cell to become a regulatory T cell. In humans, it's not so sufficient. It requires some other master regulators as well. But in order to make the cell regulatory. But long story short, it's a very, very important gene for regulatory T cell physiology, and the gene is called FOXP3, FOXP3. Mutations in this gene lead to severe autoimmune conditions, and this has been discovered by Ramsdell and Brankov, and also further after those discoveries, the gene was yet more studied by Shimon Sakaguchi and by the group of Alexander Rudensky in New York. And eventually we learned really a lot about how indeed FOXP3 induces the regulatory T cells and how it controls their immunosuppressive function and so on. So the knowledge about regulatory T cells is extremely important for our understanding of how autoimmune diseases appear and develop. And very much important for our understanding of how T cell population gets exhausted during some chronic conditions like cancer, like long-term infections, and so on. Those T cells, the regulatory T cells, potentially have a lot of therapeutic applications as well. So it's a truly massively expanding area with lots of puzzles and mysteries even until now. And it's of course super interesting to study that. I don't know, just that was. You're welcome to consider this lecture as a teaser. You're very welcome to give your feedback. You may indicate whether this topic is interesting to you, and then I will think of making, I don't know, maybe a couple lectures that that would go step by step throughout those proofs and throughout the research that led to our understanding of this area. And maybe I will say, of course, more about what mechanisms the regulatory T cells utilize in order to convey their immunosuppressive function, in order to suppress other cells, pathological cells, and so on. So, yeah, I guess for today that's it. Please set your likes there, indicate if the video was interesting, write me comments, and follow the channel. Thank you very much for attention. I hope it was interesting. See you next time. Bye.